So what we do is we build a biological hybrid computing system that is comprised of living tissue, so neurons, brain cells, that we grow into networks. And we grow these networks on top of silicon chips. At any one point in time, we're making predictions about the world. And the whole point of a biological system is to get very good at predicting the world and reducing the amount of what we call information surprise. Remember, they don't know anything about vectors or matrices. All they understand is a spike or not a spike. We go back to the idea that all biological systems are trying to fit their internal model to the observed external world. What is the external world if there is no information? It means that you are trying to fit to a null distribution.
Hey everyone, welcome Technocrats. Today we have Dr. Hon Weng Chong, founder and CEO
Hello.
of Cortical Labs, which is a company building the world's first biohybrid computing platform. The first time we met Hon was actually in Hong Kong for a conference, and you told me about what you were doing.
Yep.
I think you were working on mice cells at the time. Like this is a
Yeah,
couple
yeah.
of years ago.
That was the initial like biological source where we were. They're known as primary culture. So what we do is we grow them from mice embryos. Now, having said that, it is a very um, tedious and unpleasant process. So we decided to do stem cells later on.
Yeah, that makes sense. At the time, you showed me some stuff on your phone. I didn't really understand it. Like, my brain was just not big enough to compute what you were
Yep.
talking about. But just like a kid who gets beat up by a bully and then comes back with his bigger, buffer brother, I brought my
Yep.
younger, smarter brother with me to, like, hopefully decipher what you're talking about. So I don't feel as intimidated this time around talking to you.
No
So it should be
worries.
okay.
I hope that's what
That's
makes me
fine.
the AJ.
Yeah, it's you, man. Who else would it be? There's no one else here. I thought we could get straight into it. Like you've done a lot of cool stuff with Cleany Cloud and a bunch of other stuff. Like you're a doctor as well. So you went from the medical practice into entrepreneurship. I think really quickly, maybe talk about the transition from medical field to becoming an entrepreneur because that's I feel like a lot more people are doing that these days. So what's that transition been like?
So that's actually been quite a while now since I'd made that transition. So let's see, let's go back. So I graduated medicine. So I did undergraduate medicine to begin with. So that was straight out of high school. That was a 6-year course at the University of Melbourne. So started in 2006. So I finished in 2012. But in the last year of medical school in 2011, I was convinced by a mate of mine who was in DC, I think, around that time. So in the final years of medical school, they encourage you to do an elective. And I did my elective at Johns Hopkins in neonatology. And I was hanging out with my mate Sai, who was a developer evangelist at Microsoft. And he's like, hey, there's a special like competition called the Imagine Cup, which is designed for students. And he's like, since you're a medical student, you still qualify. And he's like, and you're doing medicine, so obviously it fits into the criteria of using technology to solve some of the world's toughest problems. Why don't you enter it and you'll have a very good chance of winning? And I had to think about it. It's like, you know what? Maybe I'll do that. So in the final year, I sat down and I came up with a couple of ideas. And just before my elective, done a unit in pediatric global health. And it was actually quite surprising to me to learn that pneumonia, childhood pneumonia, was the, I think, single largest killer of children under the age of 5 in the developing world. And the process of diagnosing it is actually quite straightforward if you know how to do it. And if you catch the pneumonia early, you can actually treat it with antibiotics, and it goes back to normal. So the issue was always the inexperience of diagnosing pneumonia and it being late. So that was the foundation of the idea. And I said, one of the ways you could actually diagnose pneumonia is just counting breaths, respiratory rate. And that's actually one of the more sensitive measures. So I said, well, why don't we try to come up with a way of automating this? Because we do this regularly with a stethoscope we place on the chest. We just count the number of breaths in 15 seconds multiplied by 4. What if we had a device which could record breath sounds and build a machine learning algorithm to count that? And then we would have a very low cost effective measure of respiratory rate that had very good inter-observer reliability. And that was the idea of Settled Cloud that was born. So I entered into the Imagine Cup with that idea. And and it's kind of funny in hindsight because one of the appeals of doing the Imagine Cup was that if you won the local event, Microsoft would fly you to wherever the Imagine Cup was being held free of charge. Then, as a student, you're like, yeah, I get to travel for free. This is awesome. Turned out we won the Australian segment, only to find out that the finals was happening in Sydney. So the free flight was just a domestic flight up to Sydney kind of thing. Anyway, so we didn't really win that one. I think we came second or third. But then what happens is they also then opened it up to a second round for the Magic Cup grants. And actually, we won a $75,000 grant from Microsoft to continue on with the work. And I took that money and put it into a research program where we employed a research nurse, and we did it with the Children's Hospital where we were testing out the efficacy of this digital stethoscope, this dinky little device I built. And so finished medical school, working as an intern. In this side, I was building the back end systems and the app and all that stuff. And one of our research nurses came up to me and said, "Hey, do you know I'm getting a lot of inquiries from parents about this device?" I was like, "What kind of inquiries?" She said, "A lot of them were asking how much does it cost and where can they buy these things." And you know, when you're an entrepreneur, you think about these kind of things. You're like, wait a minute. A lot of people are asking how much does it cost and where do they buy this? And it's a prototype device. And I said, well, it sounds like this might have legs as an idea. So I convinced my co-founder at the time, Andrew, to quit his job at Bain. So he was also a medical student, but he never practiced. He went straight to Bain. I put medicine on hold. We both packed our bags and we went over to the Bay Area. And along the way, people in the Bay Area were like, "Hey, you're here in March. You should go down to South by Southwest." And we did exactly that. We went to South by Southwest, and it's kind of wild because when we were in the Bay Area, we're actually living in one of those hacker hostel home kind of things. What is it like? 10 grown men in bunk beds all trying to chase this like Silicon Valley dream kind of thing. It was so wacky. I can't believe we actually did that in hindsight. Anyway, so we went to South by Southwest and met a whole bunch of really cool people and took the advice of not doing anything during the day because you needed a rest because all the deals were done at night during the parties. And came back with some money from angel investors, put that into building a prototype, and got something out the door, which then led to a self starter where we raised $5 million US dollars on a really great uncapped convertible note from Tencent exploration team and Ping An Ventures to turn the idea into a product. And we actually went all the way. We actually got FDAC certification and sold the devices into Amazon, Best Buy, and a bunch of other places. We really couldn't quite crack it. And this was way back in 2014, 2015, before MedTech was a thing. So that's my origin story, I guess.
Yo, is there like a chip on your shoulder? Like, what's your villain arc? Like, is there a reason you're doing all of this? Is it a humanitarian thing? Are you being a hero, or did someone like push you over as a kid and you're trying to get back at them? Like, what's why are you doing this stuff?
Oh, you mean that stuff or this stuff? Because two different
Let's
things.
talk about this current company.
Yep. So we fast forward to say 2016. So we had built a product and we had spent about 2 years, 2.5 years marketing it and selling it. We were selling into, these retail places. The B2C model was not scaling fast enough. And as you know, if you take venture, you have to grow and grow at a fairly reasonable and fast rate. We couldn't satisfy that, so we went to a B2B model. We're trying to engage with employers and employee health programs and such. That gave us a little bit of a boost, but not big enough for us to actually really go any further. And so Andrew, my co-founder, and I decided that we were going to put all our eggs in one basket and just go hunt the elephant. And we partnered up with a subsidiary from Intel, where we then managed to secure a deal with the VA, the Veterans Administration in the US. It was a huge deal for us because they put an order in for 10,000 units. And if anyone does hardware, you know that minimum order quantities from a contract manufacturer usually starts at 10,000 orders. Only problem was this was negotiated towards the end of 2016, and we thought, yep, we made it. And in November, Trump gets elected, and that's when everything went down south. Because in 2017 onwards, everybody, including the VA, froze the discretionary expenditure budget because they didn't know where the money was coming from that following year because it was either Trump got rid of Obamacare and replaced it with Trump Care, but nobody knew what Trump Care looked like, or that Trump Care would be repealed and Obamacare would come back. But there was a lot of uncertainty, and so we were just burning runway for nearly a year until 2018 when we realized we had burned so much runway that we didn't have the ability to build new product or pivot or do anything meaningful. So Andrew, my co-founder, left the company in my hands, and I had to figure out what I was going to do with it.
This is
Um,
Cloud, right?
this was Clinic Cloud,
Okay,
yeah.
yeah.
So I decided that it wasn't worth it. I closed down Clinic Cloud, and I wanted to do something that didn't rely on market forces that would be on our control, like the government. Something that was in the AI space, but I didn't want to do another convolutional neural net for X startup because there were plenty of them back in 2018, just as we have plenty of LLM companies today. But all of them were all dependent on data, and no one had more data than the big tech guys. So you were kind of at the mercy. If you were successful, they would kind of own you. So I read a paper that was written by Demis Hassabis from DeepMind and published in Neuron, and he called for the machine learning AI community to re-engage with the neuroscience community, and it did exactly that. So I spoke to the guys from the Florey Institute for Neuroscience here in Melbourne, and I asked them, "What's exciting in your world?" And say, "Hey, we have this device where we can read electrical activity, we can stimulate them." I was like, "Wow, this is really cool. How come no one's ever built like a thing where you can get it to compute, right? Because if these neurons eventually, are things that would turn into a brain, and we know that the brain computes, surely they must have some ability to do some kind of self-computation." So that was the seed and genesis of the idea. What I liked about it was no one was looking at this, so we had a unique perspective, unique angle. B, big tech hates biology, so you were not at their mercy if they wanted to like get into your space. And C, it was not tied to any government regulation or budgets or whatever. It was pure science. It would work or it wouldn't work. Very simple as that. So those were the three things that kind of got me over the line. I was like, look, you know what? I'm just going to go ahead and just pursue this and see how far I go with it. So that was when Cortical Labs was born in about late 2018, early 2019.
I'm always super interested in founder stories where a founder comes from one industry and goes into another industry. I mean, did you have any background in neuroscience or AI before taking that leap, and how did you find that leap?
Yeah, so I mean, I guess I had some background in neuroscience. Being a medical student, we all have to learn some neuroscience in order to train in neurology. So I knew a bit of that. Machine learning, I was already doing that in 2017, 2016. Actually, I probably have bragging rights for the first core ML repo on GitHub, where I put it out in, I think, what are you now? 2023, 2017 or 2016, just after WWDC, where I showed how you can actually use TensorFlow to train an MNIST classifier for digits, compress that down, turn that model into weights that you would load into Core ML, and you could do the inference on an iOS device. Suffice to say, I tried to teach this to like the Cocoa Heads community. I was like, guys, this is going to be the next big thing. It's going to be awesome. And everyone's like, oh, it's too hard. I have to learn Python. Oh, I have to learn about machine learning and all this matrix multiplication stuff's too hard. Just want an API call. And yep, they kind of ignored it. And actually, that was kind of the sort of a very eye opening moment because we were, Andy Kitchen, who's like my co-founder at Cortical Labs. We were approached by, I think, O'Reilly or somebody like that to write a book about this. And when I heard the feedback, I was like, I don't think this was going to sell very well because none of these developers really wanted to do anything in machine learning. And I was right because the impetus that we see all this AI stuff today isn't actually app developers learning about how to build neural nets, is just calling an API call. So anyway, that's a bit of a sidetrack, but yeah, I already
So in
knew.
some sense, it was kind of like a merging of skills that you were already reasonably well familiar with.
Yeah, pretty much, exactly.
So at this point, we should probably actually directly ask, what
Yep.
does your company do? What's the
What do
elevator
we?
pitch?
So what we do is we build a biological hybrid computing system that is comprised of living tissue, so neurons, brain cells, that we grow into networks, and we grow these networks on top of silicon chips which can pick up the activity of these neurons. So neurons will communicate between one another using electrical signals because they're conductive. We can see that. And we can also write to them by giving them a bit of a stimulus. So we have read-write access to a biological layer. And what my company has shown is that given the right stimulus, the right training protocol, you can get these neurons to actually inhibit a simulation world, get fed information of what's happening in the simulation world, and make actions that affect that simulation world to satisfy our requirements as the experimenter, the researcher. So, yeah, I mean,
So to give
if
a
you watch
super
enough,
simplified view, it's basically like a neural network, but actually built with neurons, as opposed
correct,
to simulated neurons.
exactly. And if you watch enough sci-fi, you'll see a very interesting similarity to one of the most popular sci-fi movies in the last decade or so.
Okay,
What movie is that?
so
Hey,
What movie is that?
is this real? Is this not? Is it a simulation? Is it not a simulation? I don't want to name things, but
Oh, fair. Okay.
I've given you enough of a hint to know whether it's the red pill or the blue pill to come to that conclusion. But yeah, that can be a little bit weird for some people.
yeah, it makes sense. So going back to the technical side of things, so I can understand how computing works because you have transistors and then you can build logic gates off that, and that makes sense. And you start getting computing, and you go higher and higher in abstraction. Whereas if you think about the biology of neurons, like you have an action potential, like an electrical signal that travels along these neurons. What are you actually writing and reading to these neurons to actually do things? Like, is it as low level as building and door gates, or are you feeding high level information and it gives you high level information back? And if so, how does that even work?
Yeah, it's not a computer. That's
Okay.
one thing, and this is the problem that we don't actually even have the nomenclature all the way of the language to express this. Another reason why LLMs are great, but when you have nothing to describe it with, that's where your thing fails. So how we do it is we don't have logic gates, we don't have any of this stuff. We also don't have a training and inference phase. It's the same loop. But what we do is we use some ideas derived from system neuroscience. For instance, ideas such as rate and place coding, right? So I'll give you an example. In a mouse or even a cat, you have whiskers. And a whisker, each individual whisker is it has a topological map of it. So it has position. And so that's place coding. That each whisker is wired to slightly different parts of the brain where it's interpreted as a spatial map. And then on the secondary thing, a whisker also, depending on where you bend it, will actually transmit frequency data differently. So the tip of the whisker will have a lower rate as opposed to really close by. So that's where a mouse can actually build a three-dimensional view of the world, right? Just by having whiskers around, by having, hitting objects around it. So it can tell if there's a corner or not, just by the differential frequencies of all the various whiskers as it hits the side of the wall. So we use these ideas to do a way of encoding the position of the paddle in the game of Pong and the ball, where we say the paddle is going to be represented in the y-axis by several places where we stimulate the neurons, and we encode the position of the ball to the paddle in the x-axis distance-wise by the rate of stimulation. So the closer the ball gets, the faster we stimulate it. So now you have XY information. You can theoretically actually, if you assuming the neurons can do Pythagoras, can figure out exactly where the trajectory of the vector of the ball is because you have rise over run. And that's the idea behind it, how we encode the information in. And all we do is we feed that information into the neurons in a specific part of the network, and we wait a short period of time and we sample another part of the connected so these neurons are all fully connected, another part of the network, and we say, okay, if there's more activity in one, we will move the pedal up. If there's more activity in the other one, we'll move the pedal down. And we do this over and over again. And within 5 minutes, they actually improve their performance about moving the paddle up to hit the ball. Oh, actually, I have to give that a caveat. You can only do that if you provide it with a reward function, kind of like reinforcement learning. And it turns out that the way we are able to reward them is by making the amount of information entropy lower in one side versus another, or one action versus another. So if they hit the ball, which is a good thing, we reward them with a sine wave, right? Because a sine wave has very low entropy, right? From one period, you can calculate n number of periods going forward. On the other hand, if they miss the paddle and they hit the back wall, we give them random white noise because white noise has infinite entropy, right? You can't calculate any sequence given any period. So yeah, that's essentially how we get
Why does
into.
that work?
Why does it work? Okay, so there is a theoretical framework called the free energy principle that was developed by Professor Carl Friston at UCL. Now, it's very complicated. There's a lot of math. I'm going to simplify it. That framework posits that all biological systems, including you and I, or a cat or a dog, or whatever, we're made of systems or distributions separated by a statistical barrier called the Markov blanket. Now, to give you an example, what it means is that when you or I are perceiving the world right now, rather than the traditional neuroscience idea of a photon hits the back of my retina, stimulates some part of the visual cortex, and it goes to a motor function, I do something. It flips the model on its head and says that we are actually Bayesian predictive machines. At any one point in time, we're making predictions about the world. And the whole point of a biological system is to get very good at predicting the world and reducing the amount of what we call information surprise. Now, information surprise, if you've done any machine learning, can be very simply like calculated as like a KL divergence between, say, two distributions. So if you had, let's say, one distribution that is like, say, the real world on one side, let's say this is like one bell curve here, and my brain or my neurons have a distribution or belief of the world, and let's say it's here. The delta between the two distributions is known as information surprise, KL divergence, or whatever. And all we're trying to do is to get very good at predicting the world so that our predictions overlap and match the real world, because that's when you get very good at predicting the world and you have no surprise, right? You have no surprise when you can accurately predict. And so if we say, take the idea of these neurons, because they're not an artificial neural network, we don't have the ability to change the weights, but we do put them into an environment where we can actually change the environment. If they do a good thing, i.e., move the paddle to hit the ball, the world becomes more predictable.
Mm.
Therefore, they're getting closer to actually being a good
So
predictor.
these neurons are constantly adapting to try and come up with some way of behaving that's going to make the world maximally predictable to them. And by giving them
Right.
sine waves when they're doing the right thing, they're going to adapt towards doing the right thing because that, artificially, but that's unbeknownst to these neurons,
Yep.
the world suddenly becomes more predictable. A nice, consistent sine wave versus random white
Yep.
noise.
Exactly.
That's really, really, really cool.
Yeah.
But one of the other questions I had was, how much do you care about like actually trying to mimic the natural behavior of these neurons? Obviously there's a lot of work that happens in organoids, and people show that the three-dimensional layout of neurons makes a big difference. But those are projects that are really trying to actually simulate the human brain more for medical studies as opposed to what you guys are doing. How much do you have to try and mimic like natural brain structure when you're working with these neurons versus you don't really care about that as long as they're alive and they're talking to each other, you guys can do what you need to do.
Yeah. I mean, it's more the latter than the former, right? So for us, it's about a getting it to do something. That was always the first objective, right? Because nobody knew or believed that you could get these cells to do an intelligence task. It's funny because people say like, yeah, that's obvious. I'm like, it wasn't actually when we started in 2019. It is now because of our work and other groups that have now started to follow this. Now, we don't necessarily need to actually uh, recapitulate everything that we know about how functional connectivity happens in the brain and so forth, right? Because at the end of the day, intelligence, and I believe in embodied intelligence, is actually a function of the embodiment of the system. These things have a completely different embodiment in the sense of they're in a virtual world. They're a paddle in the game world kind of thing, playing this game of Pong, which is not the real world. And so for us, it's less about trying to replicate all of that stuff, but try to get an effect from that. And then from there, use that as a baseline to try and perturb it to see if the perturbations we do on these systems also translate to in vivo systems. So that's one of the core areas of focus for the company now that we've shown that you can actually do these things is how closely do they correlate with the person? right? Because once you have that, you now have a very powerful analog to do drug development, personalized medicine, and so forth.
Yeah, that was very much going to be my next question: is why do I care about neurons
Mm.
playing pong? Where does this go,
Yeah.
and what are the end game application? So the end game application being like drug discovery.
Yeah, that's one of it. I mean, it's a base platform. You can do multiple things with it. So, for instance, the way the drug discovery would work is, say, for instance, and we've shown this actually quite impressively in our internally in our experiments. If you take cells from a patient with epilepsy and you grow them in a dish, the cells also have seizures, like the patient, and you can treat these cells with drugs, and they will also respond accordingly. Now, the thing here is to also know, to think about it, is which is that when you do test a drug, what you need to do is you need to test on the functional component of what the organ is doing. And for all the other organs, we have very good measures for that. Like say cardiac cells, you can measure contractility. For kidney cells, or renal cells, you can measure filtration. But the brain's functionality is not to send spikes or to fire activity. The brain's primary function is to compute. Is to cognate or whatever, right? And getting them to play Pong is actually a way of seeing how well they cognate or compute. And so, we see that with various different drugs we give to these cells, we have different results in the gameplay performance, which can be theorized as a analog or a proxy for cognition. The same can be said when we put ethanol into the cells. We know that there's a little bit of a bump of the performance, but then after a significant amount of ethanol we put into it, it just like crashes and it just dies eventually. So these are all the things that we're looking at to say, right, they're not exactly a brain, but they have enough parameters and properties for us to start experimenting with that we will find them useful, because we don't really want to recreate the whole thing, right? The last thing we want to do is make a conscious thing that's in a dish or in a jar kind of stuff. What we want to do is use it so that we can actually find answers and solutions to very hard problems, like the problem of dementia, the problem of aging, and so forth.
Yeah, so you're really
Um,
looking at trying to come up with a quantitative measure for intelligence that you can experiment
correct.
with liberally
Yeah.
without having to worry about the ethics of working on animals or working on humans or working on an accidentally conscious brain in a jar. One of the, I guess, challenges that you guys must be facing there is how far away is that from being practical? Deep tech is obviously a lot of really deep research. That's the name. And you guys started in
Mm.
2019. You've been operating for 4 years. How far are you away? Do you think from getting to a place where you're going to be operating like a business, bringing in revenue, actually starting to commercialize this work? And what drove the decision to turn it into a company when you did versus letting it incubate more in an academic environment first?
So I'll answer the last one first. It never incubated in an academic environment. We got no grants at all. This was all funded from venture from day zero, which is a problem because the moment you do that, the clock starts ticking, and eventually you do need to commercialize it. It's one of the sad facts of the matter, which is that despite discovery grants and all these things, our discovery grants actually are very iterative. They don't seem to do very blue sky, different idea kind of thing. So in hindsight, no organization, no government organization would fund this. MRFF wouldn't fund this. NHMRC wouldn't fund this. ARC wouldn't fund this either. And then you ask the question, well, what are they going to fund then? So we kind of you know raised capital from day zero to try and do this, and it was on the smallest budget, like a fifth of what you would get in an MRFF or an HMRC grant kind of thing. So that's, I guess, answers the question, why did we not incubate it? Because we couldn't, and we had to go straight to doing this. And it's, I think, a very interesting model that there are some investors out there like Blackbird who are willing to take a bet for something that may take a little bit longer to incubate, rather than having a hockey stick within like 6 months of an MVP kind of thing. What was the first question? I guess that kind of, yeah, answers the first question as well, doesn't it?
Yeah, can we move back to the tech side of things? I just wanted to fully understand. You were saying that you're reading and writing to the cells, right?
Mm.
So the computer itself is motherboard. You have a life support system for these cells, and then you also had a brain powered chip, which I didn't fully understand either. But how are you actually converting these digital signals from a motherboard to reading and writing to these cells exactly?
Yeah, so that's where an FPGA is really handy. So an FPGA allows us to do very fast, almost real-time computation
What is a
based on
FPGA?
it's a field programmable gated array. It's specialized kind of chip that can be anything. So you have logic gates that you can just program out in the field, hence the name. And so we can essentially reprogram hardware. And those hardware are then wired up to very specialized ADC, DACs, so analog digital converters, digital analog converters. So we can read analog signals and write back analog from digital. So one of the hardest things that we do, I spend a lot of time on actually, is this interface between analog and digital, right? Because at the end of the day, everything in the world is analog, despite the fact that we, a lot of it is, we have digital systems and so forth. Digital is a bit of a hack, right? We're always sampling the world, and we have questions like what the sample rate is, and people talk about what's the resolutions and AK vision or whatever. It actually doesn't, because everything is a spectrum when it comes to an analog system. So anyway, that's how we do it. So we have a system on a module that is connected to an FPGA, and the FPGA converts signals that we get from the. So we sample analog electrical signals. We then digitize them, and then from the digitization of our responses, we then feed it back to the neurons. So that's how we kind of do it.
And how exactly are you sampling the signals from the cells? Are you connecting to, this might be a silly
Yeah.
question, but are you connecting to a specific neuron, or are you like, is it in a solution suspended and then bringing signals back and forth? Like, what does that look like?
Let me see if I can show you guys
Okay.
something. So the I mean, we have a lab down there, but I'll have to go and take the thing out and show you. But here's a video that I prepared earlier. Here we go. Can you see my screen?
Yeah. Yeah.
Yeah. Cool. So this is me in my cameo role of play, play. So that's an incubator. Neurons are kept in the incubator until they mature and they have electrical activity. Um, this is a new kind of chip. Well, it's a new old kind of chip that we're using that is made of glass. And on the perimeter of the chip, there are these pads that you can see. Each pad is connected to a trace that originates in the center portion of the glass well. And each white dot that you can see there is a neural cluster. And so they grow on top of these electrodes.
Right. Interesting.
Yep. And the pink fluid is the artificial cerebral spinal fluid that we use to nourish the cells, to keep them alive, keep them healthy. It's a medium for exchanging waste and nutrients and so forth. I'm just gonna go past it. So this is how we feed them, but not that cool. All right, so here we go. This is what they look like under a microscope. So that's this thing here is a neural cluster. Here's another neural cluster. Here are these like pads that we can sample the electrical activity from, and these traces that go all the way out to the perimeters that we can then interface with our system and sample the electrical activity and also deliver a bit of a jolt or a stimulus back to give them. So yeah, this is pretty cool. So this is actually one cluster there. So there are probably like several hundred thousand, several maybe 10 or 20,000 neurons there, and and they form projections that will reach out and connect with other clusters, and they do this naturally. We don't actually stop share tell them where to grow or how to grow. They just do it themselves. What causes them to change their growth is the information, the structured information landscape that we give them, i.e., are we showing them the game of Pong? Are we showing them the game of Jumpy Dinosaur, and so forth?
Yeah, that makes sense.
So
If
when you're giving
anyone's
them
if
games,
anyone's listening, by the way, you should go check out the YouTube channel because I don't want to try and explain what I just saw, but it looked very futuristic. So just go check out the YouTube channel and you can see the video and Han explaining it. Anyway, go on, Long.
so when you give them different games to play and you're giving them different reward signals, you're seeing these clusters on each chip form in different ways.
Yep. They will. They will break off connections and they will physically move to a different location and form new connections.
And is that in response to whether or not they're getting sine waves or white noise, or is it in more in response to like the actual underlying structure of the game?
Yeah, yeah. It's to different games will give you different patterns. Different cultures will also have different patterns,
Yeah, that
but
was
the
kind of going to be my next question. Is like, do you see different performance when you work with neurodegenerated cells? Do they differentiate between different disease types? Like, do you see
we
different games playing at different levels of skill?
yeah, we do. Even from clone cells, even clusters that are identical genetically, we think that there are gene expression differences that cause them to actually have different performance. But they all kind of end up learning how to play, but they all take slightly different routes to do that. It's kind of like you and I learning how to play Pong. We all have slightly different ways of learning how to do it, but we all kind of eventually get to a point of playing it, some better than others. So it's a lot more organic. It's not really a machine, right? It's more biological.
Some
There a
people,
big difference between mice and human cells? Like, are we smarter than mice?
Yes, we are smarter than
Nice.
mice based on our data.
It's good to know.
Actually, let me see if I have this. Let me show you some stuff. I know
if
this
you
is a
take some of
podcast,
my
but
brain cells and some of AJ's brain cells and then make them compete in a
Yeah,
game of
yeah,
Pong and see
yeah,
which one of us is smarter.
yeah.
I mean, what we really want to do is we want to get some Zuck cells. We want to do some Elon cells, and we'll get them to do Mortal Kombat since they chickened out when it came to the cage match.
I
Yeah,
would
we could
sponsor
do
that
that.
research.
Yeah, it's for science. We want to know who's the better fighter. Anyway, so I kid about that bit. Let's see. So I know this is something that your listeners can't see, and it's
That's
a
fine. Force them
visual
onto YouTube.
thing.
It'll be good.
But here we go. I'm going to share with you something that's kind of cool. So this was actually at NeurIPS last year. What we did was we did a head-to-head comparison of our system against three reinforcement learning algorithms: DQN, which is your AlphaGo, Q from the Q learning. If any of your audience is like, oh my god, Q star learning, yeah, don't worry, that's an old stuff. A2C and PPO. And what we did was we said, okay, we're going to give the same game that we give our neurons, but we're going to do a very important thing, which is we're going to sample constrain them. What do I mean by sample constrain? It means that when we train reinforcement learning systems, we actually cheat because we don't do them in the real world in real time constraints. We usually have systems that run at 300 to 500 times speed up. So there's a massive acceleration beyond what's physically possible. And also, we create many more instances than we can actually have, which is not what you really want if your system is constrained by real world constraints, i.e., like robotics. So we said, since we only play 20 minutes of the game with our neurons and they're at 100 frames per second, that's 100 times 60 times 20, which will give you the total number of frames. And we capped it at that same thing. And what was really fascinating is to see that if you give the neurons, sorry, if you get the reinforcement learning agents an image input, which has a lot more information than, say, what the neurons are getting, which is only purely a ball position versus the image, even with that, the biological system outperforms these algorithms. So the one to look at here, I'm just going to zoom in a bit, is the blue and orange. So blue is MCC, mouse cortical cells, and HCC, human cortical cells. And the other three are the RL agents. And this is measuring things like average hits per rally. So how long can they keep the ball in play before missing? Number of aces, which is a thing that you want to minimize over time, which is how much has it learned to predict the first ball hit so you don't get knocked out to begin with, and then long rallies, how long can they keep the game going. Yeah, so it turns out as we sorry, this is image input, this is paddle and ball input, and finally this is the full head-to-head comparison, which is the ball position only input. And this is really important here because it shows that these systems are learning in real time, that the RL agents aren't able to do that because they need so much more data in order to process. And it's the same critique as well with LLMs. Like they're only very good because we fed them the entirety of the internet, which is, I don't know, the entirety of human knowledge in over the last 30 years or so. Quite impossible for any one human to ingest at any one time. So, yeah,
I guess
Interesting
one
side.
question that immediately makes me think is what's the energy consumption of those units? So obviously, like one of you could frame it as a disadvantage that you need to feed the entirety of the internet to an LLM, but you could also kind of maybe frame it as an advantage, is that the particular structure of an LLM is such that it is amenable to reading the entire internet, which is something that a human will never be able to do. What's the energy consumption like for keeping one of those chips alive and so that it can play the game versus keeping a simple a simple reinforcement learning algorithm alive?
Well, I mean, it's a good question because life support system, I think it uses about like 20 watts maximum of power. The neurons themselves use 10 to the minus 4, minus 5 watts of energy for 800,000 to a million neurons. It's not surprising. The human brain only uses 20 watts of energy for like billions of neurons, right? So, energy consumption wise, massive difference. And to give you a perspective, to put it into an apples to apples perspective, that's like saying I got my pocket calculator with a solar panel on it to learn how to play Pong or play an RL game, which we know is impossible because we only can do them with very large GPUs that suck up 250 watts minimum just pass it through the silicon die. Not to mention all the extra bit of cooling that we have to put around it to prevent it from melting down, right? So there is the additional, not just the CPU, the GPU cooler on top of it, but there's the environmental cooling, right? The air conditioning that you need to get these things to actually not overheat, kind of thing.
And what are some of those challenges when you're working with actual neurons? Like, how do you guys think about like evolutionary divergence with your cell cultures over time? Do you keep going back to the stem cells and regrowing them? You talked about there's a divergence between clones. When you're working with cells, getting the experiment right, or not right, but consistent is a huge challenge. Like, has that been challenging for you guys, and how did you overcome?
Yeah, it's always a challenge, right? And the only way, and this is it, unfortunately, with biology and with most of the sciences, you just need a large sample size to reduce the amount of random observations, right? And that's the reason why our experiments take longer. They're a lot more rigorous, right? Because we are forced to do things like collect 10,000 hours of gameplay, do 250 samples minimum kind of thing in order to show statistical significance. Now, I haven't seen very many papers out there. I guess, machine learning is one of those things where no one reports how many random seeds or how many parameter tweaks that were happening. They just report the best result as a frozen weights kind of thing. And so I don't know if you've ever tried replicating any of these papers, but it's very hard to actually train anything, let alone if they don't provide you any weights. You have no idea whether it actually works or not.
What does scaling look like in this scenario? So I think you guys are at 8,000 brain cells right now, like neurons sort of connected. Is there a
Yep.
limitation of how many
800,000.
you can actually have in parallel connected to each other, or is there some other thing? Because I know in like traditional AI, it's going to be parameters and compute as like the way that you get more intelligent over time. What does that look like for you guys?
I'm not too worried about scaling that part yet. My biggest worry actually is in the input output. How do we represent matrices, tensors, and vectors to these neurons? Remember, they don't know anything about vectors or matrices. All they understand is a spike or not a spike. So they're analog systems. And so we're trying to focus right now on how do we convert the analog to digital and digital back to analog in a more efficient way, which is trying to do it in the time domain that we don't really seem to be using very effectively. And so, yeah, the analogy I make is I'm less worried about scaling how big the neuronal system can get because it's equivalent to saying I have a server farm of H100s, but I'm connected to that server farm with a 56K modem. You're not going to be able to do too much with that until you've reduced the bottleneck and get a really fat 10 gigabit per second like Ethernet connection to it.
Is 8,000 kind of the tip off point right now that makes sense for you guys to use, or like smaller or bigger? Like what? Why that particular number?
Oh, that was just the number we did because we just wanted to get something to work, and we
Okay.
just noticed that the bigger the network got, the better they performed, and so we just threw everything at the wall. We haven't done ablation studies yet, so we don't really know what the lower limit is. It could very well be that we only need maybe a tenth of those cells, and they could still play the game. But we're a privately funded lab with limited runway, so we have to pick our battles accordingly. But we would love if more people started looking into this. using our systems.
And so, is that the main battle that you guys are fighting right now? Is thinking about how to improve that I/O interface, or is that like what is the primary thing that you guys are focused on today?
Yeah, yeah. So we're trying to do that, and the primary thing we're trying to focus on as well is to, and part of our commercialization is, can we make systems that allow other people to experiment on our device, so that they can come up with new ideas and hypotheses of why these things are working or not working, right? We want
Would that
people
be buying
to
physical kits, or would there be some way of you guys having the equivalent of a cloud infrastructure?
Yeah, it's gonna be both. So if you have a lab and you can grow cells, you can buy our kit and you can just grow them and use our system to interface with them. But if you don't have a lab, we can grow it for you. You just give us your code and we run it for you to get your results back, and we give it back to you, kind of
And
thing.
what does code look like for one of these things? Is it just
Yeah,
FPGA code and like a three-dimensional layout?
no, no, no. Well, it is, but that's the reason why development takes so long and it's hard is because we are building abstraction layers on the back of the FPGA, on the back of the C kernel, the libraries, and so forth, so that you can program these things in Python or JavaScript. Or really not program them, but program the environments that they're in and create the rewards and punishment signals and the particular waveforms that you want to stimulate these neurons in a high level language so that you are not rate limited by your knowledge of a low level language and shuffling memory around, but you're limited by the amount of creativity and the way you can express your ideas in that language.
So really what you're doing is you're kind of programming the lab environment. Uh,
Correct. We're building the CUDA, so to speak, for the neurons.
yep, very cool.
Should we look into the ethics of this? Because things are gonna, like, if they get bigger and bigger. I do know that you've talked about the difference between sentience and consciousness before. Do you want to quickly touch upon that? What the differences are?
Yeah, I mean, it differs from different people, but we've defined it and we put our this is our stance on it is that there is a difference between sentience and consciousness. Some people use the terms it's interchangeably, but I'm like, why would you have two words to mean the same thing, right? You really want to have a distinction. So the way we think about it is that sentience is a way of responding to a stimulus, right? So to give you an example, a amoeba or a tadpole or whatever we would consider them sentient, right? Because you poke them, they don't like it, they swim away. You put some food there, they like it, they swim to it, kind of thing. Are they conscious? I doubt that an amoeba is conscious. Maybe they are if you are panpsychic or whatever. So that I think is where we draw a line. Consciousness, on the other hand, requires a higher level of cognitive, I think, capability and unique evolutionary traits. So mostly, I think a lot of the social animals have consciousness. Some people would say dogs are conscious. Maybe even cats, crows, humans, dolphins, elephants, those kind of things. I don't know if a chicken is really conscious, so to speak. So, consciousness requires a lot more ability to know you exist and that others exist. Even like children, I don't think they're actually conscious until they reach theory of mind stage at 2 or 3 years old, kind of thing. Because in most cases they don't view themselves as an individual thing. They feel themselves as the extension of their caregiver, usually mom or something like that. That's the reason why kids will usually cry when mom cries and are happy when mom's happy, kind of thing.
So I mean, that's an interesting, like, I guess, partitioning of the two definitions. Do you think that shifts the ethical line, though? Like, to you, where's the ethical line? If a child is not conscious, it's still wrong to perform certain experiments on them.
It's a good question, and I think this is something we can't really define it because it's a societal construct, right? Ethics ethics itself is a societal construct, and if it's a societal construct, it means it's fluid and it changes based on the values of society at any one point in time. The reason I say that is, you can think about it this way: IVF babies, right? Oh no, you're not allowed to mess with God's, way of procreating or something like that. Today, it's like, yep, it's subsidized under Medicare because it brings a lot of joy and happiness to couples and so forth. Values will change, and I think this is a conversation that needs to be had with society in general. At what point do we think that lines have to be drawn, and at what point do we redraw those lines as we learn more about the technology and how we use it and what the benefits and the harms are?
Yeah, I mean, I definitely think I'm certainly don't fall into the camp of trying to preempt ethical dilemmas when technology is still so nascent that I don't think those conversations are really worth having yet. But do you, as your company grows and succeeds and achieves, new scales and new techniques, do you anticipate headwind from the general public? I mean, like the general public is already like tearing itself apart seemingly around LLMs. Are they conscious? And I think that one's very easily answerable at this stage, and people are still freaking out about it. And practically speaking, it creates headwinds for artificial intelligence development. Do you see those
Hmm.
headwinds? I mean, surely they're going to be coming your way.
No, 100%. And that's the reason why we already engage with bioethicists. We have papers out with bioethicists looking at, the use of this kind of technology. We 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 come from a different world from the AI people. Let's put it this way. AI people come from computer science land. And in computer science land, there are no regulations. It's a free for all. And hence the reason why the culture has always been more of let's not ask for permission, but just seek forgiveness, right? That's the Facebook mantra, the hacker mentality. When you come from the life sciences, you can't do that because we all come from a space where if we make a therapeutic there that goes into a person, there are ethical and moral concerns of will this person get better? Can you make claims that are unbacked and so forth, right? So there is a higher barrier, a higher hurdle that we have to cross. For instance, all our work here have to go through ethics and institutional review boards, right? It takes time, and this reason why it's a much harder endeavor than say building an LLM, because all you have to do is just scrape the internet and just feed it into a very large GPU. Don't need to ask permission from anybody for that. I guess it's changing now. You have to ask for copyright permission and so forth. But ChatGPT was made without any of those considerations in mind. So I think we are in a different space, and we do think quite heavily about the moral and ethical questions about this. Now, this is a thing that comes up quite regularly in cases of like, in bioethics questions of like the principle of double doctrine, where you then have to weigh the merits and the cons of any intervention, right? Because for instance, if let's say one of the pathways to getting a dementia cure may be needing to create semi-conscious or partially conscious entities in order to test them. Is there a moral or is there an ethical net benefit for creating these things versus the risk? Some people may say yes, if you live with dementia or you're a carer for somebody, because that could, you know, save someone's life and save a family or a person a lot of agony. But others may not, right? Because you are creating something that is conscious and so forth. It's the same question we have with animal studies. Is it ethical to test drugs on primates, right, for safety and efficacy? And I guess as a society, we have said yes because these lead to life-saving drugs. So it's one of those things that we have to think about and try to navigate the landscape, but not by ourselves, but as a conversation
Yeah,
with
it's
society as a whole.
I'm really curious. How does
Yep.
one even go about measuring whether or not something is conscious? Like, are there standard practices in your world where people can, you know, actually even begin to attempt to quantify the chance that this cluster of brain cells is slightly more conscious than that cluster of brain cells?
No, not really. Unfortunately, it's still a field that has a lot of debate. People are very partisan about it. Nobody really, there's no even set or fixed nomenclature to actually talk about these things. What is consciousness? What's sentience? How do you define these things? What are the parameters? Where do you cross the line between sentient to consciousness? Poorly defined. There are people like David Chalmers who've spent his entire lifetime and career trying to work these things out. But there's only a handful of people. A, because it's not very sexy, right? It's mostly philosophers, it's mostly bioethicists that work in this space, as opposed to a mad rush to commercialize a piece of technology like an LLM. So it's a field that is slower because there's less funding and there's less glamour attached to it, but very important, I think, for a field like ours or even the AI space to start considering these things.
So on that note, how much do we actually understand of what's happening inside of the cluster of cells? Like, if we're sending input output, is it sort of like a black box? So you're like sending stuff in and you don't really know what's happening underlying, or do we have a good understanding of what's happening at a cell level or even a signal level?
I think we don't have definitive understanding of what's going on. We have ideas, primarily also because it takes a long time and a lot of resources to hone the exact mechanism of action. But one idea that I have, and I think that it needs to be formally validated, is that part of the part of the reward stimulus being a predictable sine wave or a predictable signal lends it to the fact that there may actually be a Hebbian plasticity event that happens. If you think about it, it makes sense, right? Intuitively, if you have two very predictable signals happening, there is a higher likelihood or chance that they will actually overlap and that they will do it regularly at a regular period, which then means that you have now a Hebbian event as opposed to random signals that have very little, regularity in their overlaps. So, that could be one explanation for it. We know that Hebbian plasticity leads to long-term potentiation, which causes synapses to form. But then again, we need to look into this in more detail. Why this is happening, and we wish that there was a little bit more funding, more grant funding, actually, to look at this in a more, I guess, exploratory, basic science kind of question. And then that I think is completely unknown, brand new field of science. That is just waiting to be explored.
And what about the memory side of things? Like, is it remembering things? Like, and if you retest it or a couple of different trials, like, what does that look like?
So we don't know. We so the way we do our experiments, we have multiple protocols. Sorry, we have a protocol where we have multiple runs of the game, and we don't do it for more than 20 minutes because the chips that we were previously using generate heat, and it causes a disruption in the osmolarity of the cell culture media, and that's very detrimental to the cells. And so, what happens is that we once they learn how to play the game, we take them out and we put them back in the incubator. We put it back in, and it looks like they lose their ability to play, like they forget. But the question is, are they actually forgetting, or have they adapted to a new information landscape, which is a null information landscape, because they're not getting any signal at all, right? And if we go back to the idea that all biological systems are trying to fit their internal model to the observed external world, what is the external world if there is no information? It means that you are trying to fit to a null distribution. So we think that rather than them actually, quote, forgetting, they're actually just adapting to a null information landscape.
Is there a difference? Like, if you put me in an empty room for a year, I'd probably forget a lot of things.
I think if I put you in an empty room, you probably go insane within 48 hours. I mean, we know that that's a torture technique the CIA uses, just sensory deprivation. And it's almost impossible for us to think about it, right? Even when you're sleeping, you're still receiving information.
I guess that was kind of the tongue-in-cheek point, right? Is that, like, it's
Mm,
forgetting, not just being placed into a different environment where what you were remembering is now no longer relevant. And like, I don't remember
correct.
half the stuff I was taught in primary school because I'm not being quizzed on it every day, right?
Exactly, right? So this is a big open question. Are they forgetting, or are they just, zeroing out because that's what they're trying to adapt to? And it's a very unnatural state. No system, no thing that we know of actually goes into a no information, like, state kind of thing. Um, and it's quite interesting as well, because if you don't actually feed these neurons information, eventually over time they actually end up becoming oscillators. And once they start oscillating as a pattern, they are impossible to train. You have to throw them away and start from scratch.
In
Yes.
some sense, like, they can become useless.
Yeah, pretty much. They form the connection so strongly that it's very hard to break the oscillatory pattern. But it makes sense.
It's
Oscillators
like
are the naturally
sprinkling
lowest
some
energy state.
LSD on them and seeing if they can lay new pathways.
Maybe we need to put some DMT or something like that and tell them to snap out of it.
Yeah.
I was gonna ask, do you know where the stem cells actually come from? Like, which human they would come from, and I guess on top of that, then can you draw correlations that, like, this particular human's cells does better at Pong than this one, or this one does better at this particular thing? Is there, like, specialization and happening at that level, like, for humans?
So a good question. We don't know for a lot of themselves because they're from anonymized donors. Before we had the ability to grow our own. Now that we have ability to grow our own, some of the cells are actually my cells.
Wow. Okay.
Yeah.
So are you doing just your cells, or are you getting a couple of team members and then having them compete and, like, seeing what the results look like from person to person?
We have myself and another one, I can't say who,
Okay.
but we've donated ourselves and also donated our bodies as guinea pigs. Because at the end of the day, what we really want to know is, if we do a study, if we do a trial, if we give a certain compound to the cells and we see an activity in that, and we feed a control, blinded control on our cells, on the person, and the compound, do we get the same response? Now that would be really, really like powerful if we can actually show that there is a correlation between what happens in vitro and what happens in vivo, because that means you no longer need to test drugs on yourself; you just test it on an analog.
Yeah, that's awesome. I'm looking forward to the future where like I go to MSY and instead of like AMD versus Intel, it's just like I can get a Loong chip or a Hon chip, or like I can just pick the person I want.
You take some from us for sure.
Yeah.
Yeah, I mean that way you can say when you're bored at home, you're like, you know what I'm gonna do? I'm gonna play with myself tonight. You can like literally have a co-op game with yourself.
Yeah,
So,
yeah.
so one thing I think that's probably worth mentioning for people who are listening in is that even existing animal models, like when you put cycler or like any sort of like CNS based drug into mice or primates, the correlation with humans is already like still very bad. And one of the other
Yep.
key challenges is that you're dealing with live systems, and so how do you even give a mouse Alzheimer's? This is something that
Mm.
takes a really, really long time. And so there's a standard way, there's a couple of labs that are certified to do this. They create standard mice models, and then you have to give them different standardized versions of various diseases. And even if you're getting that process perfect, which we are far from, that takes years because you have to wait essentially for this neurodegeneration effect to take place, especially if it's not a genetic cause. And this just takes a huge amount of time. It's part of the reason why the CNS field moves so slowly. Is that with cancer, you can give something cancer and then see whether your drug helps. But it's very difficult to give something a neurodegenerative disease, and it's very very difficult to do that in a consistent way. And so it's just worth pointing that out for people who are listening who aren't familiar with the field, because these chips, if you can create a correlation between what's happening on these chips and what happens in humans, not only is that potentially a much better correlation because it's just a fundamentally different type of technology, and you could be using human cells that can even be personalized to the people that you're anticipating doing clinical trials with, but they will also just be dramatically faster to create. And I think that's one of the big potentials for brain on chip like technologies. Part of the
one Hundred
reason I'm
percent.
excited for it.
100%. You summarize it really well, right? Because if you think about it, reason why we don't have more drugs in this space is it's a 1% success rate. It's really low at the current way things are
There's
going.
less
Um,
0.1% success rate depending on where you draw the starting line.
Correct.
But yeah,
Exactly, right.
terrible.
It's very hard, with even for instance a translational mice model kind of thing. So we've been working with some companies that can literally CRISPR in conditions, and we thought we were going to have something really cool with this disease called Progeria, which is, I don't know, like you may be familiar with Benjamin Button, right? The very old creepy looking kids, they just have accelerated aging. And we've realized that the Progeria that we CRISPR into mice do give them this accelerated aging, but we don't see that in human cells.
It's not dissimilar from
So
Alzheimer's, where all of that mouse Alzheimer's models are the genetic variant, which is only 5% of the Alzheimer's that you see in humans. I think this is a really
yep.
good place to wrap it up, on that note of like where this technology goes and how it actually fundamentally affects humans. Because I think when you start with something like playing Pong, there's just a
Mm,
question of why bother? And when that
yeah.
comes all the way back around to helping solve some of the biggest challenges in the drug discovery and biotechnology space, which is models that are translatable and aren't full live animals and are fast and are cheap. I think that's a really nice full circle, and gives people a clear picture of like why should we care about this kind of technology. So Hon, I just wanted to thank you for coming on and sharing that with us today. It's like I learned tons. I've read a number of articles about how these kinds of brain on chip systems work, but I've never been able to dive deep with someone who's an expert in the space.
No worries. Yeah, thanks for having me on the show.